So let's go down the list. First, I want to kind of review I'm going to hit the highlights. The last video I sent you really did hit the highlights of DNA, so I don't want to go over that again. It's uploaded already on the YouTube channel, so please review it. That's the one thing I forgot to say on the last video. We do have an exam next week. So let's make sure you understand that. Let's start with number one here, is that there is an exam next week. And what is that exam going to be on? It's going to be on biomolecules and biochemistry. All right, so you really do have to understand the things that we're talking about, and there will be at least 2 quizzes next week. Now, there's an interesting point here with the quizzes, all right, and the test. And there will be one lab. So you have one exam, 2 quizzes, right? And you'll have 1 lab all next week. That all will be on the first quarter, right? Yeah, because so that also means that none of this can be made up. None of this can be retaken. Does that make sense? So you got to get it right the first time, because this is it. I mean, I got to put the grades in Friday. So biomolecules and biochemistry is for next week, and that has to happen. I should have mentioned that in the last video. All right, so what did we go over so far? I'm going to go ahead and summarize this. The DNA stuff, watch the previous posting on the YouTube channel, right? It's like a half an hour, 34 minutes long. You don't have to watch the whole thing, just watch what you need. It does go over everything it summarizes everything we talked about, from ribose all the way up to the double-stranded and the anti-parallel direction of the DNA strands and how the biochemistry of DNA works. We did talk already about sugars, right? Carbohydrates. And we posted that already, right? Everybody at least saw that much. Let me go to another slide here. With the carbohydrates, what's important? I think a lot of kids always want to know what's important. You got to remember that in most classes you have to figure out what's important, and you can figure it out by listening to the teacher repeat themselves over and over again. I made a chart for you with all the different monosaccharides, right? And you had the polysaccharides. Who remember what a saccharide is? So the saccharides are things like, polysaccharides would be things like what? Glycogen. So glucose would be with monosaccharides, but good guess. Other monosaccharides would be what? Galactose. Sounds like a lot of people have to study. And another, the last monosaccharide, one of the last monosaccharides was which one? Fructose. And all of these were what? Yeah, they were sugars, but they were also known as hexoses. These are all. There was one monosaccharide that we talked about that was very important, that led us into discussing DNA, right, and RNA. That was the pentose that we know as what? Hold on a second. The pentose we called what? Well, the one we called ribose. And there were 2 forms of ribose. The ribose that took that C, that C blank H blank O blank, right? You remember that? C blank H blank O blank? You don't?
Oh yeah. Oh, I 1, 2, 1.
That's right, 1, 2, 1. And ribose is a pentose. That means what? 5 carbons, right? So 5 carbons. What about the hydrogens? 10. 10, and then the oxygens? 5. So that's that's ribose. Now deoxyribose had the same formula, except what? Had 5 carbons, 10 hydrogens, and 4 oxygens. So this is the exception. This is an exception to that rule about sugars, because and it's an exception. They tell you it's an exception by changing the name to deoxy. So don't confuse that. Remember, ribose looks like a pentagon, very much like fructose does, except fructose has 6 carbons. And here's the oxygen, and then there's a carbon that comes up here. This is 1, 2, 3, 4, and then 5. There's your 5 carbons. This is the place, I believe yesterday I made a mistake and I pointed to this carbon. That's not correct. It's this carbon is the place where you either have an oxygen or not. All right, so if you have an oxygen, it's ribose. You don't have an oxygen, it's deoxyribose. The 3', these are all primes. Remember that? They have a little tick mark, the little apostrophe. If it's a prime, 3' is where it connects to the other sugar below it. The 5' is where it connects to the 3', right? 5' connects to 3'. Do you remember that? Yeah. All right, so if not, it's all in that video. I'm not going to go over the details. I just wanted to remind you what ribose is because it's such an important sugar. So the polysaccharides, we talked about glycogen. What else did we talk about with polysaccharides? Glycogen, cellulose. What else did we talk about with polysaccharides? Say again? Say again? Tell me. Chitin, good. And let's spell it right this time. Chitin, right? I'm sorry I misspelled it last time. So each of these, there's cellulose, there's chitin, there's glycogen, there's a couple more. What are they? Starch is right. And what's the next one? Amylose and amylopectin are forms of starch, so that's good. So it would be amylose and amylopectin. What's the one that's missing here? So let's go over the list. This one's in the animals. No, not yet. That's in animals. This one's in plants. This one's in plants. This one's in fungi and in insects or arthropods, right? Amylopectin, that's right. Amylopectin and amylose are starch. Peptidoglycan. Peptidoglycan is found in what? In bacteria, right? So that's the polysaccharide. We're talking about a polysaccharide that we find in each of these different categories, right? Also, we know that starch and glycogen are for what? Storage of energy. Excellent. And that includes amylose. That includes amylose, of course, right? And that. Why is it doing that? Let it go. And it includes amylose and amylopectin. But what is the, then when we look at cellulose, when we look at cellulose, chitin, and peptidoglycan, what do we know about those? They're for structure. They're for structure. Are we all okay with that?
Did you say the cellulose is for plants and the chitin is for the go up?
All right, say that again.
The cellulose is for plants, right?
Cellulose is for plants, that's right.
And what's the chitin for?
You tell me. Do you remember the chart? It's posted on the YouTube video, so I'm not gonna, I don't wanna spend a lot of time going over it. I'll say it quickly, even though I think you'd be better if you looked it up yourself. I think you'd learn it more deeply. So I'll say the answer, but I encourage you strongly to go back and review it and put on index cards. Chitin is for the exoskeleton of arthropods, including insects and lobsters and shellfish of all kinds. Arachnids, all of them have chitin in their exoskeleton, and chitin is also found where? In the cell wall of fungi. All right. Fungus. We talked about mushrooms, et cetera, right? So that's all the polysaccharide stuff I want to talk about. We have to get moving here. Then we talked about the disaccharides. Remember, we talked about some examples of disaccharides. And disaccharides, we said there were two kinds, right? Or there are at least two that we discussed. One was lactose, right? And the other one was what?
Table sugar, but I forgot.
Very good. It was table sugar. It's called sucrose. Lactose had two monosaccharides. It had glucose, and we kind of do it like this. And it had lactose also has galactose. And they're connected. I shouldn't have drawn it that way. They're connected by an oxygen together. So this galactose and glucose, they form a disaccharide found in where? Found in milk. Limited use for adults, much more useful for babies. That's why we make cheese out of it, because cheese is easier for us to digest than milk. But even so, cheese is not. Some people that are lactose intolerant can't eat it because it upsets their stomachs. Sucrose is what is glucose again. And then what? Sugar. That's right, fructose. Now, I'm not sure that's right. I think it actually might be, I'm not sure where the connection is between the two. But in any case, the fructose and the glucose, that's sucrose, that's table sugar. All right? That's it. That's all. That's pretty much a review of all the carbohydrate stuff. And the details, the sugars, these disaccharide discussions you will not find in the video that I posted before, nor the one I posted last night. Again, we lost that information. If you need a one-on-one tutoring session on some of this, you can come and see me, make an appointment. I gave you the chart. You took notes in it in class. This stuff is all in the previous video. There's several charts that have it. So just make those index cards and memorize those things. So now we've covered, let's go over again one more time really quickly, a big summary of what we've done so far. You see how I keep reducing? We had all those details and we just keep reducing. The first video was an hour and 57 minutes, and the video I posted last night was 35 minutes. No, I think if you watch yesterday's, it summarizes the DNA stuff, but you should watch the previous video and just skim it and find what you need out of it. I'll try to make chapters this afternoon. But if it's a very on-task, quick question, go ahead.
No, it was on, yeah. I was saying, are we going to need to know schnapps? Like,
Yeah, for sure. Yeah, you got to know all that stuff. Schnapps. What elements, schnapps, what are the elements in biomolecules, right? What are elements of life? Schnapps, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur.
Is that
What are the elements? What are the elements in fats? Carbon and hydrogen. What are the elements in carbohydrates? Carbon, hydrogen, oxygen. What are the elements in nucleic acids? Everything. All the schnapps, right? What are the elements in proteins? Everything but phosphorus. Very good. And so you get this idea that things, that's the stuff you're going to see on a test. And that's why I want to give you a couple quizzes before you take the test, and give you a couple activities next week before you take the test in a lab, and let you play around with these ideas. But it's not going to be very useful if you haven't gone over and tried to memorize these things before next week, because next week, Wednesday, as of right now, it's Wednesday, is your test.
I was just about to ask, is our test Thursday?
I'm thinking Wednesday. It might be pushed back to Thursday. I don't want to leave it to Thursday because Friday I got to put the grades in. Yeah. All right, so today I'd like to look at proteins, and then tomorrow fats. Fats are going to be kind of the introduction into the cells. So let's look at proteins. Proteins are polymers as well. They can be dimers. They're usually called peptides if they're small, really tiny. But let's take a look at the monomer of proteins. So what is the monomer? What's the monomer of carbohydrates? What are some monomers of carbohydrates? We just went over them. Huh?
What was the question?
What's the monomer of carbohydrates? The monomer. Glucose, very good. Fructose, galactose, right? Those are carbohydrate monomers. You could have a ribose, I guess. I'm not sure, but usually ribose is involved in the. All right, so those are the monomers of carbohydrates. What are the monomers of nucleic acids? Hold on a second. So monomer of proteins are called amino acids. Amino acids have a very, very specific structure, and they look like this. They have C, N, H2. There's that nitrogen that we talked about, right? There's nitrogen here. NH2. There's an H sticking out here. How many bonds does carbon make? 4. So they need 2 more. We're going to use this. We're going to write this R. This R is not an element. There's no element on the periodic table named R. Thank you. Some dust or something here is making my nose itch. It's crazy. Could you give me some tissue, please? Give me the box. Thank you. I appreciate you helping me out here. All right. So we have R, and R is a variable. It's a variable. So it can be up to 20, R can be 20 different amino acids. Different, as you say, side chains. All right, maybe 20 different ones. And I'm gonna draw a few of them. I'm not going to draw it all 20.
You guys say NH2?
Yeah, this is NH2. This is NH2. Is that not clear? Do you want me to rewrite it? Let me rewrite it. Is that better? So it's NH2. And then out on this side is C double bond, double O, OH. So what that looks like, by the way, COOH looks like this: a carbon connected to an oxygen is connected to a hydrogen. But how many more bonds do I have here? How many more bonds do I need for carbon? 2 more. And so what you get is a double bond O. See the double bond? Do you see that? There's 2 bonds to this oxygen. This structure, this is known as a carboxyl group. It's also known as a carboxyl. The whole thing would be a car.
You said there's 2 bonds on hydrogen, that thing that, what is that thing, the COH? What did you say that was?
So it forms an acid. So carboxyl group makes a carboxylic acid. So what you have is this thing's acidic, so it can release this hydrogen. This hydrogen can go off, it can leave. And when it leaves, it releases hydrogen ions, and that makes it an acid. So this part here is the acid part of the word amino acid. This part here, this NH2, this is the amino group. So the amino group's there, and the acid part is here. So that's where you get amino acid. Is that clear? Is that somewhat clear? Well, I wrote it down already, but the amino part is this NH2, you see it? And then the acid part is a C double bond OH, called carboxyl group or carboxylic acid. All right?
Is that an A carbox or a carbox?
Car with C-A-R. So it's an acidic carboxyl group, right? So it's an acid. Now, do you really need to know the group names? I used to actually ask the kids to memorize some really important groups. I don't really think it's as useful as I used to think. I think if you just know that when you see something that looks like this, you know it's an amino acid. If it helps you to know that that's an acid, this is a carboxyl group and that's a carboxylic acid now. Or this is an amino group, so that it helps you remember that this is an amino acid. If that helps, great. But as long as you can recognize that this is an amino acid, can you tell the difference between this and a sugar and a nucleotide? I hope so. Because that's what you're going to have to be able to do. You will see some of these structures on the test next week. So do you have to actually draw it the way I just drew it? No, you don't have to draw it. But you should know what these things are, what they look like in general, be able to identify them. So when you have 2 amino acids, they come together. Here's your NH2. I'm going to draw it out. Instead of putting NH2, I'm just going to put actually N and then the H2. Then the carbon. Then there's going to be this R group sticking out here. Well, why is the R group up, Mr. Mendoza, instead of down? Why are there 2 H's instead of 1? Those are minor things right now. Just know that there are. I'm sorry, my nose is just out of control. There's something going on. Something's in bloom. It will be in the video forever, unfortunately. Some of the videos I recorded, especially there was one time I had bad allergies, and all you heard is me sniffing and sucking up snot, and it was gross. Well, you never had to say all that. I like the way you said that. All right, so here's a hydroxyl group. I told you what a hydroxyl group was again, and then there's that double bond O. So that's a carboxyl group. So notice it looks different the way I drew it out here, but that's all right. I think it can survive. And I did that wrong, didn't I? Because what did I do? How many bonds does this have?
5.
Is that okay?
No.
No, I messed up, didn't I? Yeah. It's more than that double bond. What am I missing? I'm missing a whole carbon. So that might be a way that might be, and that was not on purpose, that was an accident, but that might be a really good way for me to test whether you know that the carbon has 4 bonds. If I put an amino acid with a carbon with 5 bonds, Are you gonna be able to identify it's wrong? That might be a good way to do it. I think a lot of people will, I think a lot of people won't. So when two amino acids are next to each other, under the right conditions, and with these things called enzymes, somebody mentioned transcription earlier, this is what happens. It happens with the enzyme that's called, a ribosome. And when you have that enzyme, a ribosome, and you have these two or more of these things sit next to each other, what happens is this reaction occurs. This hydrogen and this hydroxyl group, they leave, and what happens to them? What do they make? What do they make?
Water.
Yeah, water. There you go. Somebody's thinking. So out comes water, H2O. Out comes H2O. But then what, all right, so you got water out, but why did it leave? How did that help anything? What was the end result? You got water out, but what happened to those two monomers? What did they become? What did they become? You still got your double bond O, that's still there, isn't it? Isn't that still there? Isn't that there? Isn't it there? Yeah, these three are gone. They're in water, they're gone. Bye. And what you're left with is now this, and this has one hydrogen. This is missing a bond now, isn't it? Nitrogen is missing a bond. Used to have 3, now only has 2. And is anything else missing a bond? That carbon, yeah, that carbon at the end is missing a bond too. So let's check it out. This one has 4 bonds, we're good. This has 2, 1 to the hydrogen, 1 to the carbon, that's fine. This oxygen has 2, that's good. This carbon has 4 bonds, no problem. This R group, we don't know what's going on, but that's fine, it's a variable. Here's H, this 1 to H, that's good. Nitrogen only has 2. It used to have 3, 1, 2, 3. Now it only has 2. This one has 1, 2, 3. It used to have 4, now it only has 3. How do you solve this problem? They bond to each other, right? They bond to each other. And this thing here, this is called a peptide bond. It's a type of bond, and why? It's just sharing electrons, the same as everything else that we talked about. The only difference is that it's between C double bond O and this amino end of the group here, this amino group and this carboxyl group. When they share a bond, we call that a peptide bond. That helps us recognize that this is now a protein, it's a polymer, right? It's starting to become a polymer. This is not a polymer yet, this is just a dimer, right? This is only 2, just like a disaccharide, right? Do you agree? All right, so this is actually not a full polymer yet. It will become one, or should become one, but as of now, it is just this. And when it continues to happen, though, once you start getting rid of these over and over again, when this goes away again, this OH goes away, and you get this double bond O, and then it's bonded to another protein, another amino acid, 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 another amino acid. You get this thing called a protein. Now these circles represent what? What do these circles represent? One monomer. One monomer. So remember, a monomer is an amino acid. A monomer is amino acid, and so this is one amino acid, so this would be amino acid 1. And then here's another amino acid. This is amino acid 2, right? Then there would be 3 and 4 and 5 and 6 and 7, 8, 9, 10, 11, 12, 13. Right? So it's a long chain of amino acids connected by a peptide bond, and we call that a protein. A protein can be a bunch of these chains kind of put together as well. So we like to call this a polypeptide. Right? A polypeptide is this thing that just is a long chain. There's 20 amino acids. You know, again, when I first started teaching, I had the kids memorize what all 20 amino acids were. I had to learn in college. I thought, why not let them do it here to help them? Maybe, maybe not. But I find that it's not as useful as I thought. And I'll include a picture of it in the final video. But take a look for a second at what this looks like. These are the 20 amino acids. So that R group that sticks out, they could be leucine. These are the 20 amino acids that you find in protein. These are what you need to eat every day. Not every day, but every so enough of all of these amino acids in order to be healthy. You can find them in different places, by the way. It's not just from meat. Some people think it has to be from meat. You can get all 20 amino acids from different sources, but you need all 20. Some of these you can make yourself. Some you can't. There's some essential amino acids you need, but some you can't. A lot of these you can. And you can see that some of these have not. Can you see this? Can I zoom in? I wonder if I can zoom in. Do you see it? Oh yeah, you see. I can see clearly these things. There's a bunch of amine groups, CCH3, nitrogen. There's nitrogen in these things. Some of them have rings. Some of these are pretty big, aren't there? Tryptophan, which a lot of this is in turkey. Some people like to say it's what causes us to get sleepy after we eat turkey. This is a really big amino acid arc chain, isn't it? 2 double rings, and then there's a long chain. There's a real tiny one. The smallest of these is which one? Which was the smallest one? Look up there and tell me which is the smallest amino acid. Yeah, the first one, alanine. Alanine is the smallest amino acid. Now you might see all these different colors. And I'm going to send you this, I think, because even though you don't have to memorize them, it's really neat that you get to. I'm going to give you an activity where you're going to have to decide what amino acid goes in which protein. You don't have to memorize the shapes, right? I don't expect you to be able to look at this and tell me it's leucine, or look at this one and tell me it's isoleucine, or look at this and say it's proline, or look at this and say it's an alanine. I don't expect you to do that. But I do expect you to tell you if I have a protein that's full of a lot of sulfur, then I expect that you're gonna tell me that it must have what 2 amino acids in it? Aspartic acid or glutamic acid, right? Because those are the 2 that have what? Sulfur, right? Oh, I'm sorry, I didn't mean those 2. I meant cysteine and methionine. Do you agree? So cysteine and methionine have sulfur, so I expect you to be able to tell me that. If I tell you which ones have sulfur, I don't expect you to know that they have sulfur in it, memorize it, no. But if I tell you, hey, which amino acids are in this acidic protein, this protein that is very acidic, what two amino acids should have those 2? What 2 amino acids should be in that protein? One or both of the amino acids. These 2, right? Aspartic acid and glutamic acid should be in it. What if I tell you a protein is basic? Arginine or histidine, right? And so there are qualities to the amino acids that change the protein. So I know there's something that people love to talk about. I personally don't understand it, but let's talk about it. Let's talk about hair. Hair is a protein. Did you know that hair was a protein?
Yes.
Okay, so hair is a protein. And so again, these are the 20 amino acids that make up that R group, just to remind you. Hair, people love to talk about hair. And all it is just a bunch of amino acids, one after another, after another, after another, after another, after another, all et cetera, et cetera, et cetera. And then they wind around each other, they kind of braid around each other like this. And a bunch of these molecules braided around each other, these polypeptides, but they make a single hair. Is that clear? So this single hair is made of a lot of amino acids. And it sits next to another hair, doesn't it? Because you have a lot of hairs on your head, hopefully, unless you're bald. So you have two hairs. There's a lot of amino acids in it. So sometimes these hairs, they curl around each other. They do this. They curl on themselves, right? Some of those curls are tight. Some are looser, right? Some hair is just kind of wavy that tracks each other, but it isn't quite as loose. And some is just plain straight. They don't attract each other at all. So why do these proteins do that? Well, it turns out that if you zoomed in, the amino acids here, this one has, or actually I should draw it over here, so let me draw it in yellow. This amino acid and this amino acid contain sulfur. So maybe this one's a C, cysteine, right? And this is methionine, M. And what they do is they create this disulfide bond. So they actually attract each other and link. They cross-link. And they hold each other tight, so you get tight curls. If you have a lot of these, in the protein that you're making, then what happens? It's going to be really curly. Does that make sense?
Mm-hmm.
And so the hair curls up real nice and tight.
So putting heat to it breaks it apart.
Good. We're going to get to that. Excellent. These bonds are like hydrogen bonds in that they are not bonds, they're not covalent. So the bonds that hold this together, this hair together, they're covalent bonds. They're very strong. It takes a lot to break those bonds. These bonds are a lot weaker, just like hydrogen bonds. So remember that as we go and discuss. So of course, when the hair is curly but not as curly, there's less of these, right? So instead of having it every so often, you have a little less often, and they curl. They still curl, but just not as tight. And with wavy ones, there's one here, maybe there's one all the way over here. They want to attract each other, but they don't really do a good job at it. So these attractive forces, there's less and less of these. And of course, the straight hair probably has very little sulfur in it, right? Make sense?
Yes.
So which amino acid you have in the protein, what does that mean? Can anybody tell me? What does it mean? All this discussion about hair, it's not really about hair, right? What does this mean? What's the meaning of all this? What's important about it? Why is this important? How can you generalize? Yes, it's true. The more sulfur you have, the more sulfur amino acids, the more methionine and cysteine you have, the curlier your hair. Okay. Is that really important? I mean, I guess to some people, but why is it such a revelation? Why is it such a big deal? What does it tell us about how things work?
Amino acid changes them?
Excellent. Yeah, that's it. Amino acids, the sequence of amino acids, what the order is, which is not only the quality of the amino acids, how many of those types you have, and the order in which they are to give you a different phenotype. These are phenotypes, aren't they?
Yes.
These are phenotypes. These are traits. Curly hair, straight hair, light skin, dark skin, tall, short, fat, skinny, green, yellow, wrinkled, smooth. Yeah.
So would let's say you have curly hair, right? And you put heat on it, and then you get so pieces are really, really straight and other pieces are curly. Is that the breaking of the bonds, or is it just
All right, say that again.
So like, when you have like really curly hair and you put heat on it, you get heat damage, where it's like some strands are curly and some strands are straight. Is that breaking of the bonds?
Excellent. Excellent question. When you damage your hair, whether it be because you put the wrong chemicals on, the chemicals are too strong, or you do it too often, or you heat damage your hair because you overheated it, right? What you're doing is you are breaking the covalent bonds. You've gone too far. You see that? So now the hair falls out, or it's some of the hairs are curlier than others, and some just you just start to lose quality of the hair as a whole. Because when you look at hair, you're looking at the entire thing, not the millions of hairs that make up hair, right? Because when you look at hair, I just look at hair. So, oh, your hair looks nice. I mean everything, all the millions of strands of hair on your head. When you overheat it or put too many chemicals, the covalent bonds break. When you add just a little bit of heat, though, that's those flat irons and the chemicals, they straighten it out. What you do is you break the disulfide bonds, and then you add a chemical, that hair straightener stuff. What it is just a chemical that blocks this. So you add oils will do it too. What it'll do is you block it. If you block it, then the thing does what? What happens to the hair? It straightens out. So you block it, the hair straightens out. It's just a chemical reaction. These are just forces. These are electromagnetic forces, like little magnets. If the magnets aren't there, it's just going to lay straight. So people that have straight hair, naturally, without all these chemicals or heating or whatever, they don't have those sulfur disulfide bonds that would naturally come back together. By the way, when that chemical kind of falls off on its own, those disulfide bonds reform because the sulfur's still there. The covalent bonds were never broken. This order, the order of the amino acids and which amino acids are here, that is what we call the primary structure. That primary structure determines everything that happens after it. Because proteins do what? What do proteins do? Look at hair and tell me what proteins do. They do grow. We can grow proteins, yeah, in the case of proteins. What do? They attract. So what are they doing? What is hair doing? From one to the other. What is it doing? This is not changing. This hair didn't change. All these hairs are the same. It's doing what? Make curly. It's folding. Proteins fold. Proteins fold. And the folding makes a shape. And the shape determines the function. Now with hair, so the fold makes a shape, the shape makes a function. What's the advantage of having really curly hair versus straight hair? I don't know. You'll have to figure that out. So I'm sure somebody's looked at it, but I don't know of any. The point is that in general, when you change the shape of a protein, you're changing how it works. Like a key and a lock, right? If you have a key of a certain shape, it'll open the lock. If the key is a different shape, it won't open the lock.
Does the shape of the protein determine its porosity, like how easily something can get into it, or how easily it blocks something?
Good question. Yeah, it does. It determines a lot of things. Yes, the question: does the shape determine its porosity or the ability for something to get into it? Well, that in many cases, yeah. Some proteins are very good at absorbing things. Some proteins are very good. They do more than just absorb things, right? It's more than just being absorbent or not absorbent. Proteins actually do something. Well, let me finish talking about the structural component. But I do have something to say about that. It's a good question. So, I don't know what happened there. That's weird. Oh, I see what I did. All right, so there's primary, there's secondary, there's tertiary, there's quaternary structure, because not only do you have primary and secondary, so primary is the order, right? Whether you have arginine and histidine and isoleucine, or do you have histidine, no arginine at all, isoleucine, and maybe you have arginine. Oh, not arginine, what did I just say? Asparagine. All right. So maybe this makes the protein completely different, right? Just like we just talked about, if you have a lot of if you have the two amino acids, cysteine and methionine, then your hair is really curly. If you don't have any of that, then your hair is really straight. So you change the function of the hair, the structure of the hair, the whole thing, based on the change in the amino acids, right? So that's true that's a really basic idea that impacts everything that happens in your body, whether you fall asleep in class or not. Whether you fall asleep in class or not. All right? Sit up straight there, or go stand up in the back if you can't stay up. Go on. I know life is hard for you. Get up. So, whether you understand how you feel, how much energy you have, as I said, falling asleep in class, all that has to do with these proteins, these things we call enzymes. You've heard of this term, enzymes? And all enzymes are proteins that, not all of them are proteins, some are RNA, but they're folded in a specific shape. So this would be enzyme 1, and maybe this is enzyme 2. And what enzymes have are what are called active sites. So maybe this part right here helps a chemical reaction happen. And maybe this part and this part together make a chemical reaction happen. If your eyes are closed in this class, you're leaving. Let me be clear. I don't care if I'm not going to get an argument about if you're sleeping or not. So when you're dealing with a substrate, this is the enzyme, a substrate that looks like this, which enzyme is going to work on that substrate? Number 1 will, because you see what happens with 1. What do you notice about 1? Look at this shape and look at this shape. They fit like a puzzle piece. Excellent. So substrates are the thing that enzymes work on. Substrates are what enzymes work on. So if you had another substrate that looked like this, where would that fit into? Which enzyme would take care of that? 2. This is extremely important. What I'm telling you now is extremely important. On the end of course exam in May, I promise you there will be questions about this. In very complicated questions with experiments that ask you to change different temperatures and pHs to see which enzyme fits which substrate. I guarantee, it's in the practice OST. I'm sure it'll be in the final OST. So how enzymes work, how proteins work, is their shape. That shape determines the function. The shape of this enzyme is going to determine which substrate it acts on and how it acts on that substrate. Do I have any questions before I go on?
How did you spell safety?
Well, I really misspelled this. Let me just get rid of it. Thanks for bringing that up. Kick a guy when he's down. No, you did it right. You did the right thing. I was just joking. So substrate. All right? So the substrate. Now you guys are taking physical, right? Conceptual physics, right?
Some of us
Some of us, some of you have had some chemistry. So what, do you remember, you've seen something like this, right? A plus B yields C. Have you seen that before? So this is called a chemical reaction. It's called a chemical reaction. These are called what? Well, they would be, well, they are variables. They are made of elements. You're right on that. I'll give you a hint. This is called a product. Right? So this is a product here. What are A and B called? Well, they have a lot of different names, but usually they're called reactants. It could also be called reagents, but they're the two things that are coming together to do something. Enzymes don't get changed. They're not part of this. What enzymes do is they make this chemical reaction go faster. So these enzymes don't change. They stay around until they break down either naturally or they're broken down. These enzymes are not involved in this reaction. So if you have a bunch of A's, hold on, and you have a bunch of B's, and you're making a bunch of C's, this enzyme that looks like this makes it go faster. All right, so that's it for today. Have a good day. It's time to go. It's 11:57 exactly. Thank you. So as you see here, as we just discussed, you have enzymes that are made of proteins that speed reactions along. And they have active sites, or areas where those R groups become chemically active. They help the reaction move forward. They speed the reaction along. There are some key concepts of enzymes, and we will, again, enzymes are just one example of a function of what? Proteins. They can be of RNA as well, but functions of proteins. These proteins, they have areas where they're chemically active. In these areas here, chemical reactions occur that don't occur in other places. So you have these active sites. The enzyme acts as a collector. It allows, in this case, we can take a look and think of this enzyme as bringing A and B together. There are these things, these chemicals, these molecules are floating around. In order for A to react to B, A has to find B. Now they'll find each other eventually. And what are two things that you can do to speed up a chemical reaction? Say, you can add an enzyme, that's correct. But the word for the enzyme, if this is an enzyme, you can add an enzyme, that'll speed it up. It's going to speed it up by collecting A and B and bringing them together. And when they come together, what do? They form C, right? So the enzyme is not involved in the reaction itself. It's just bringing those components together. What it does eventually, if you think about a chemical reaction, if you think of this, let's take a look at something like this. If this is C, and this is A and B, and this is the energy of the reaction, energy required. Right. And not required, but energy, say it's free energy. If this is the amount of energy in the system, you need a certain amount of energy for A and B to come together and for that chemical reaction to occur. Once they do come together and you form C, energy is released. And these things become more stable. That's why chemical reactions occur, because they become more stable. It's sort of like if you take a ball and you put it on a hill, what does it have? A lot of potential energy or very little? If you're going to sleep, you need to get up and go on the side of the room and stand up, go to the bathroom and wash your face. But if you're closing your eyes in this classroom, you're leaving. You're not going to insult me. I'm not going to sit here and watch you sleep during this class. So let's make sure that that's happening. I know in other classes you sleep and that your teacher doesn't takes a different philosophy, that's fine. I take the philosophy that if I'm going through the trouble of sitting up here and explaining something that's going to be on an exam, I expect that you are paying attention. Here's a ball on top of the hill. Does it have a lot of potential energy or little potential energy? A lot. And so when it gets down to the bottom, does it have a lot of potential energy or little potential energy?
Little.
What happened to that energy?
It turns to kinetic.
It turned into kinetic. It was released, right? And you lost the heat, right? So some went to heat, and you also turn into kinetic. Once it's down here and it rolls out, all the energy was lost to heat, right? Agreed? Understood?
Yeah.
Here, in this example, in a chemical reaction, when you have two things that you're bringing together, one thing that you're splitting apart, whatever type of chemical reaction you're dealing with, you have something you're starting with, the reactants or reagents, and then you end up with the products. When a chemical reaction occurs, in general, there are very many specifics, but when you need to think about it this way, that when a reaction happens, the product ends up being less energy than the reactants. That's why they do it, because it's like a ball rolling downhill. They become more stable. This is why two molecules react, or atoms react with each other to form a new substance. This is why two substances react together to form a new substance, because the product is more stable. If you take gunpowder and you react it with oxygen, you get an explosion. The explosion is the release of energy. That's this part. That's this part of the reaction from here to here. That's the release of energy. Just like this was what? The release of energy. Do you see that? That the product of gum, after you explode, you get carbon, right? Have you ever seen carbon, like the powder that's left over after an explosion? You see it in movies. The stuff that burns, when you burn wood, you release energy, don't you?
Yeah.
Right? When you're burning wood or burning natural gas, you release energy in the form of light and heat. That's what we call burning. That's when you're mixing oxygen with the carbon that's in these fossil fuels. Do you agree with that? Carbon dioxide is released, energy is released. So do you agree then that the product must have less energy than what you started with? Okay. So now there are other kinds of reactions known as endothermic reactions, but we're not going to discuss those right now. The bottom line is that these reagents or the reactants have more energy than the products, so the reaction happens because it releases that energy and they're more stable. It's much harder to explode, to burn ash than it is to burn wood, right? If I took this paper and I burned it, I'd have black ash left over. Do you all agree with that?
Yes.
All I need to burn this paper is a little bit of heat, don't I? What do I need? I need a flame. I need a lighter or a match, and I can light that little tiny flame, and it'll make a big flame, won't it? That little bit of energy will release all the energy that's in this paper, or a lot of it. And what'll be left over is ash, black ash. Do you agree? What if I took that black ash and put that lighter next to the black ash? Will it light up like this did? No, because it doesn't have as much energy. The energy's gone already in the form of light and heat. Does that make sense? So again, the products have less energy than the reactants. We know this. by these examples that I just given you. But by these same examples, when you exploded that gunpowder, when you burned that paper, what did you need to add? Does gunpowder blow up on its own? Will this paper burn on its own?
No.
What do you need to do? Add energy. You need to have a little bit of energy. We call it activation energy. You need to add a little bit of energy in order for the two reactants to come and finally meet. For A to meet B. For them to meet each other and hit each other in a certain way in order to meet each other in a certain way in order to form that new substance, whatever that new substance is. So it's sort of like a pool game. If you're going to get the pool ball into the hole, what do you have to do? They have to hit each other. And you need to have energy, don't you? Mm-hmm. All right? So this is the energy required for these two, A and B, to meet each other. A and B have to meet each other. Well, what the enzyme does is it lowers that activation energy. Now, with the enzyme, it gets to it faster. And so it requires less energy. So it doesn't have to be as hot. So if this reaction, A and B, you'll see, only happens without an enzyme, the reaction needs, I don't know, make up a number, 70 degrees Celsius, all right? You have to have 70 degrees Celsius worth of energy for the A to meet B and to produce C in a timely way. And it takes an hour for all the A's and B's to meet each other and make C at 70 degrees Celsius. If I add an enzyme, it could take that same hour in less temperature to get the same results.
Can you only add one enzyme? Or like multiple?
It's a good question. You don't add one enzyme, you add a certain concentration of them. One type of enzyme, it's usually one enzyme for each reaction. So if you have a chemical reaction, it usually takes a specific enzyme to make that reaction occur. So remember we talked about these shapes? Enzyme 1 only will work on substrate, on this reagent, on this reactant. Enzyme 2 will only work on this substrate or this reactant. Does that make sense?
Yeah.
All right, so you have specific enzymes work on specific substrates or reagents or reactants. Without the enzyme, it works at this temperature. With the enzyme, less temperature, same results. Less temperature, same results. Or less time. So if you leave, you can same results at less temperature, or faster results at the higher temperature. Now why? Because the temperature, increasing the temperature already is making them meet each other faster. The enzyme just increases the speed of that meeting happens. Are we all clear on this? Another word for an enzyme is a biological catalyst. A catalyst is a substance that speeds up a reaction. It does exactly what I'm talking to you about. It lowers that activation energy. It lowers that requirement of energy for the reaction to happen. How it does it is it increases the rate at which the reagents meet each other. It increases the rate at which the reactants meet each other to form the product. The same amount of energy, as you can see, the reaction happens. This is still at the same point as it was as it is now before you add the enzyme, but after you add the enzyme, it requires less activation energy to get at that product. This difference from where A started and C ended up is still the same before or after you add the enzyme. Are we all good with this?
Yes.
All right. That's basically how an enzyme works. We'll talk more about it. You're going to see a lot of enzymes. There's going to be a lot of discussion about how enzymes work. All right, so let's take care of proteins. Now let's go ahead and talk about lipids. Let me start a new video for lipids.